The cell cycle consists of 4 phases: G1 or gap1, S or synthesis phase, G2 or gap2, and M phase. The G1, S, and G2 phases are parts of the interphase, and the M phase includes mitosis and cytokinesis. For many eukaryotic cells, a cell is duplicated every 24 hours, and mitosis takes 80 minutes. The first stage of interphase is the G1. The cell does most of their growing during this phase. It synthesizes new proteins and increases in size. After G1 comes the S phase. During this phase, the DNA in the nucleus is replicated, and at the end of this phase, the cell contains twice as much DNA as it did at the beginning. Next is the G2 phase. In this phase, the cell finishes growing and 2 centrosomes have appeared in the cytoplasm. After G2, it's the M phase, which begins with mitosis. Mitosis has 4 phases: prophase, metaphase, anaphase, and telophase. In prophase, the genetic material inside the nucleus condenses and the duplicated chromosomes become visible. At the same time, protein strands called microtubules appear from the centrosomes in animals. Then the nucleus disappears. Next, the nuclear membrane is broken down. At the same time, microtubules or spindle fibers are growing from the centrosome. These strands attach to the centromere of each sister chromatid. After prophase is metaphase. During this stage, the sister chromatids are lying along the center of the cell so that both chromatids face towards opposite poles of the cell. The spindle fibers attach to the centromere of each sister chromatid, and now the sister chromatids are ready to be separated. Anaphase is the next stage of mitosis. The sister chromatids are separated through a shortening of the microtubules attached to the centromeres. The poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the 2 ends of the cell. The last stage of mitosis is telophase. At this point, the spindle fibers are broken up. The chromosomes are surrounded by a new nuclear membrane at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. After telophase, mitosis is completed, and the next and final process is cytokinesis. During this process, the cell splits into 2 cells. In animal cells, that occurs when the cell membrane is drawn inward until the cytoplasm is pinched into 2 nearly equal parts. Now, each part contains its own nucleus and cytoplasmic organelles. This includes mitochondria, Golgi bodies, and the rough ER. Once split, the 2 new cells are now fully in the G1 phase of interphase and ready again to begin their growth. The cell growth is regulated carefully because there are very severe consequences of uncontrolled cell growth in a multicellular organism, such as cancer. Cancer cells do not respond to the signals that regulate the growth of most cells, so the cells divide uncontrollably. Cancer cells form a mass of cells called a tumor, which begins to displace normal cells and tissues. Not all tumors are cancerous, but the cancerous ones are malignant, and malignant tumors invade and destroy surrounding healthy tissue. Additionally, cancer cells are dangerous because if they enter the bloodstream or lymph vessels, the cancer moves into other parts of the body and forms other tumors, a process called metastasis.